So in this study, essentially, the reason why we wanted to explore this is because in myelodysplastic syndromes and in CMML, we know that there's a lot of epigenetic, or meaning mechanisms that regulate how our genes express that are involved in how the disease manifests. So essentially, the objective was to evaluate this compound called Seclidemstat, which is a molecule that inhibits LSD1, which is one of these molecules that regulates this process, combined with the azacitidine. So we developed this phase 1/2 clinical trial, which is currently completing the first dose escalation portion, to try and explore the efficacy and the safety of this combination in patients who have myelodysplastic syndromes and CMML and have kind of failed the standard therapy with hypomethylating agent. And so far we've reached close to completion of the phase dose escalation. So far we're seeing good safety, which was evidently important. And now we're starting to see some signs of activity, which evidently we need to wait until the dose expansion, but around 50% of patients that we've treated have had some response to therapy, which is encouraging. So obviously it's a different kind of mechanism of action that we're trying to explore to see if that could help improve the outcomes of these patients with myelodysplastic syndrome and CMML that otherwise don't really have very good prognosis. So far we've seen, as I was mentioning, around a 50% response rate, and we're just waiting to see once we complete the dose escalation and we're able to treat more patients at the same dose level in the dose expansion to see what further experience we gain with the combination.